So where we left off with photosynthesis last time was tracking the atoms in there, looking at where the atoms end up, that you could follow and see, okay, if there is 6 carbons at the start, how you end up with 6 carbons at the end, that nothing is created or destroyed, it just gets moved around in the process in there. So we used glucose to simplify the explanation in there. However, what you end up producing is a 3-carbon sugar, and then you have to glue those 3 carbon sugars together to make glucose, which is a 6-carbon sugar. And we'll go into more detail about that later on. So the simplified equation is going to show that it actually is going to consume water, which makes sense because when you do the metabolism the other way of breaking down sugar, it's going to yield a little bit of water in there. So this picture is kind of a nice one for having your overall big picture of what goes on with photosynthesis. You've got the chloroplast. So you're going to have your light energy that's going to come in, and you're going to have these light reactions that are going to generate your ATP and your NADPH. Those are going to be used to fuel the carbon cycle, or the Calvin cycle here, which is sometimes referred to as carbon fixation in there when you add in the carbons and add them on. And then you have the NADP and the ADP, which are this without the phosphate, and you're going to have the electrons moved over here into the Calvin cycle in there so that you can go back and regenerate things in there. So the start is going to require the splitting of water. So oxygen is going to be given off from the plants when they split water into the hydrogen and oxygen. So that's why we get oxygen out of the plants in there. And basically, C.B. Van Niel proposed that all photosynthetic organisms are going to require hydrogen in there. Water is the source of hydrogen for the plants. They're not using the oxygen. They just want the hydrogen from it. But there are other bacteria that are going to do photosynthetic reactions in there that don't need water to do it. So the sulfur bacteria, they're going to use hydrogen sulfide in there, and instead of producing oxygen gas, they produce sulfur gas in there. And there are several other reactions that will use something where instead of having oxygen here, it will replace it with something else in there, some other element to do a type of metabolism in there. And it gives it its source of hydrogen, and then it releases that off as a gas as well. So when we look at other metabolic cycles that are out there, that's the general pattern for it. In here, all you're really worried about is the photosynthesis in there. So it's a redox process, just like metabolism is, or respiration. In respiration, the energy is released from the sugar when the electrons are associated with the hydrogen and get transported by carriers to the oxygen. Remember, oxygen is that final electron acceptor there. In photosynthesis, the electrons are going to increase the potential energy as they move from water to sugar, and it's going to require energy from light. So with photosynthesis, you have to put energy in. With respiration, you're getting energy out. So we have 2 stages of photosynthesis. We have the light-dependent reactions and the light-independent reactions. The light-dependent reactions are going to be dependent on there being light. So they take energy from the sun, the solar energy, and will convert it to chemical energy. Your electron acceptor in there is the NADP plus in there. So the solar power from the sun is going to reduce NADP plus to NADPH in there. So that's going to be what goes back and forth. You'll have photophosphorylation in there where you're going to generate ATP. So you'll use chemiosmosis to power the addition of the phosphate onto ADP to make ATP. And then you've got the Calvin cycle that's going to incorporate carbon dioxide from the air into other organic molecules. So with the citric acid cycle, you were pulling off carbon dioxides that you're going to exhale. With the Calvin cycle, you're putting on carbon dioxides. So what's going on in photosynthesis, if you look at it, a lot of those reactions are almost the opposite of what they are in respiration. So the carbon fixation is the initial incorporation of carbon into the organic compounds. So if we look at the light reactions in more detail, we've got sunlight, which is going to be a wave in there. All light is waves. And when you look at it, you've got the electromagnetic spectrum here. The shorter wavelengths are going to be down there at the smaller numbers. The longer wavelengths are over here. They're higher energy when they are the smaller numbers. Lower energy are going to be the bigger numbers for the waves in there. So they run opposite in there. Bigger wavelength, lower energy. Shorter wavelength, higher energy in there. So visible light is just a small portion of it here that you see. The smaller waves, gamma rays, X-rays. So these types of rays can actually do a lot of damage to cells because they do have so much energy in them. And that's why you can be damaged from X-rays in there. Yeah. Like from an earthquake? Sound waves? Yeah, the sound waves are going to be way over here. Yeah, way even up on this side in there. So UV light still is able to cause some damage, but it doesn't cause as much damage to us as X-rays do. So you can have more exposure to UV radiation in there than you can to X-rays or gamma radiation without it causing damage, just because of it being lower levels in there. So then you've got infrared, microwaves, and radio waves here in there. If you are exposed to any of these for a long enough time, it can potentially cause damage with those. It's just we aren't usually exposed to them directly for large amounts of time either, so we don't worry about them in there. So visible light's just that small portion of it, from 380 to 750 nanometers. And light's kind of a strange thing. It will behave as a wave, and if you study physics in there, it's going to behave like waves do, but it will also act like a particle in there. And so we refer to a particle of light as a photon. It's not actually tangible, but it does have a fixed amount of energy in there. So our photosynthetic pigments are going to be the light receptors in there. So a pigment is able to absorb visible light. So when you're seeing something, what you're seeing is the light that is being reflected off of it. It's the light that it absorbs you don't see in there. And so different pigments will absorb different wavelengths of light. The spectrophotometer is an instrument that is used to measure the ability of a pigment to absorb various wavelengths in there. And you can come up with an absorption spectrum in there. In plants, we have three pigments in the chloroplasts in there. We have chlorophyll A that's going to participate directly in the light reactions. Chlorophyll B is an accessory pigment. And then you've got the carotenoids that are a group of accessory pigments in there. And when we look at the action spectrum in there, chlorophyll A is going to be kind of a violet. What's going to stimulate that is kind of your violet and red lights work best for that. And then you'll have different lights are going to stimulate different types of chlorophyll differently, is basically what it comes out to be in there. So when we look at our accessory pigments in here, the chlorophyll A is kind of a blue-green color in there, where chlorophyll B is more of an olive green color. So you can see on here that they're going to absorb different spectrums of light in there. And then you've got your carotenoids that are going to be your shades of yellow and orange in there. They just help to broaden the photosynthetic range of things in there. So what are some things that you can think of that are going to be high in carotenoids? Oranges, red and yellow peppers, carrots, anything that is the yellowish orange colors in there in the plant world is going to be high in the carotenoids in there. So what about when you see the leaves changing to the oranges and red right now? Why do you think we see that right now? What it is the chlorophyll is drying up in there, the chlorophyll A and B, and now we can see the carotenoids that were already in there. But the chlorophyll A and B are usually going to mask those other ones. So that's what you're seeing, is those colors that are hidden in the leaves at this time of year. But these colors, these orange and yellows, obviously don't keep it alive anymore. So another thing they do is provide some photo protection in there. It's going to help to absorb and dissipate excess light that would normally damage the chlorophyll. So plants can actually get a sunburn in there. And so there are certain plants that they say plant them in the shade. And they mean that. I've tried planting them in the sun because I was like, well, I like that plant and I want it here. And it doesn't work. It will die. It will get burnt and it will die like that. So what happens is the light is going to come in, and light's got the energy. The light energy comes in, and it's going to stimulate one of the electrons in the chlorophyll to this excited state. And when it goes into that excited state, we know that things always want to go from the higher energy state to the lower energy state. Things naturally roll downhill like that. They don't naturally roll uphill without something pushing it up the hill. The light's what's pushed it up the hill here. So it's going to naturally want to come down in there to its ground state. Some things will actually have kind of a fluorescence to them. And what causes that fluorescent is that electron falling back down to the ground state in there. And it can actually cause a visible fluorescence. So also, when you are going to have a photon absorbed, it's only going to absorb the energy that is an exact match to it. So if you have a pigment here that absorbs 680 nanometers of light, and you give it anything other than 680, it will not absorb it. It only absorbs the one that is the exact match. So that's why when I take and put those plants that want to grow in the shade and have a different spectrum of light in the sun, they're not happy. I can't just give it more water and say, here, that's maybe what you needed was a little bit more water. It needs the different spectrum of light in there. So the afterglow is just that fluorescence. So when we have these photosystems in here, it's the reaction center complex along with the light harvesting complexes. And so you've got your light harvesting complex that's going to have the various pigments in there. And then you've got the photosystem, which is a protein complex that's going to have a reaction center in there. And that reaction center is going to send up its electron to its primary electron acceptor. So this is just showing the energy coming in, moving down, being trickled through, and then pushing the electron up in energy. So we have two photosystems, and they are numbered based in the order they were discovered, not in the order that they're used. Photosystem 2 is going to be the one that functions first. And the reaction center chlorophyll A there is going to absorb a wavelength at 680 nanometers. And then your photosystem 1 uses chlorophyll A as well, but it's going to absorb at P700, or it's at 700 nanometers in there. So there's a slight difference of wavelengths that they're going to absorb. So this has what we call linear electron flow. This one uses 2 of the photosystems. And this is a lot like our electron transport chain. So you'll look and you go, okay, that kind of looks familiar. Passing electrons down here. Oh, these cytochrome things. And this is like the equivalent of your electron transport chain for plants. Except things happen backwards. You do your electron transport chain first in the plants to get the energy. So you've got your electron coming in, and then it being passed down, and then it being stimulated again through photosystem 1 here, and coming down to produce the NADP. So we refer to this as linear electron flow in here because it starts at one, it moves to the other, the electrons are moving down the line in there. What you also have is a separate final electron acceptor here, which is significant for that. So this is just a wordy explanation in there. I'm not concerned about you knowing the names of the different proteins that those electrons are passed through. Just know that with linear, it's got the 2 photosystems, and the electrons get passed to a different final electron acceptor. With the cyclic electron flow here, it uses photosystem 1 but not photosystem 2. So it skips this whole first step here. And instead, what's going to happen is you excite those electrons, goes to a primary electron acceptor, it comes back, and the electrons end up back at the original reaction center in there. So it's a cycle that fills itself in. So if you're trying to make a comparison with what you saw with respiration, you had those 2 electron carriers in there, NADH and FADH2. One kind of cut the process short, the FADH2 cut the process short. NADH passed it the whole long way. So with cyclic electron flow, you're cutting it short. With linear, you have to go through both. So what is in the gray, black, and white here is the processes you have from linear, and with cyclic, it's just the smaller amount in there. So different organisms will use different types in here. It's mostly a bacteria thing that you're going to see this cyclic electron flow in there. So when we look at the chemiosmosis in the chloroplast versus the mitochondria, both generate ATP through chemiosmosis in there, so creating that gradient separating the electrons. Some of the same electron carriers are going to be used, including the cytochromes. There is an ATP synthase complex that is similar in both of them, so that proton pump at the end is similar in there. Your source of electrons in the mitochondria comes from organic molecules. Your source of electrons in the chloroplast is going to be from water. So in mitochondria, it's used to transfer chemical energy from food to ATP. In the chloroplast, it's going to use the light energy to make ATP. So this is where you get the heterotroph versus the phototroph in there. And in mitochondria, the inner membrane pumps hydrogen to the matrix, to the inner membrane space. And in the chloroplasts, the hydrogen is going to get pumped from the stroma into the thylakoid space. So they actually, function very similar in there. So any questions on those light reactions so far? So big thing with them, you'll either have cyclic or non-cyclic in there. Cyclic uses photosystem 1, non-cyclic photosystem 2 and photosystem 1. You've got the different pigments that are going to absorb the different wavelengths of light in there. And that afterburn or fluorescence is just the electron coming back down. So you don't see that in a whole lot of things, but it is kind of cool when you see it, where you'll see it sometimes in bacteria in the ocean where they will kind of fluoresce in there, and it's that electron coming back down. Yeah. I believe there's some in the plant world that do it. I've never seen one. Yeah, I've seen it in the ocean, but I've never seen it on land in a plant. But that's something you're likely to see on a test, because I think it's pretty cool that it happens. So the Calvin cycle. So when you look at this thing, it looks a lot like the Krebs cycle or the citric acid cycle, except it's going to be gluing things back on instead of pulling them off like we do in the carbon cycle. So what you're going to have for your input in here is you're going to have 3 carbon dioxides enter at one time. This is the carbon fixation phase that's going to go on in here. And so it's going to use some of that ATP to do that you generated in those light reactions. It's also going to use some of those electrons here from the NADPH. So in phase 1, you're going to incorporate, you have 3 that come in to do this, but they're incorporated one at a time. They're attached onto the 5-carbon ribulose bisphosphate. It's abbreviated as RUBP. And then it's got an enzyme in there that's a carboxylase that they gave it its own name, Rubisco. Not really worried about you knowing that. You just want to know in phase 1 that you've got the carbon fixation in there. So it makes that 6-carbon intermediate in there that then splits into 2 3-carbon molecules. So in phase 2, you've got the reduction going on. So this is going to involve moving the electrons in there. So each of those 3 phosphoglycerates is going to receive ATP in there and lose phosphate to become glyceraldehyde 3-phosphate. So that glyceraldehyde 3-phosphate is one of the same things that you actually have in glycolysis in there. So for every 3 carbon dioxides, you get 6 glyceraldehyde 3-phosphates, but only one of them is a net gain of carbohydrates in there because it took 3 carbon dioxides to get there. So it begins with your 15 carbons as your 3 RUBPs in there. Then it goes up to 18 carbons involved with it, with 6 glucose 3-phosphate or glyceraldehyde 3-phosphates in there with 3 carbons each. 1 exits to be used by the plant, and the other 5 are going to be recycled to regenerate the 3 RUBPs in there. So the third phase is the regeneration phase in there, where you're going to spend some more ATP in there to rearrange things in there, and it can accept a new carbon dioxide again. So when you look at the picture, you just want to think you've got your carbon fixation, reduction, regeneration. So one of the things we run into in arid climates is you've got these stomata on the plants that are going to take water in, and they also are going to allow water to go out on there. So when it is hot and dry in there, you can actually have the plant start to lose its water when it's letting the carbon dioxide in there. And that's going to cause the plant to start to wilt and decrease its photosynthetic yield. So when the carbon dioxide decreases in the oxygen spaces and you begin to have more oxygen buildup in there, it's going to start a wasteful process of photorespiration in there. So in your C3 plants, the first organic product of carbon fixation is the 3 carbon compound, your 3-phosphoglycerate in there. What's going to happen is you're going to start to have the decrease in carbon dioxide in there. You're not going to be getting the carbon dioxide in there. And the rubisco is going to bind to oxygen in place of the carbon dioxide in there. And then it splits and makes 2 carbon compounds instead in there. It still consumes the ATP in the process without generating it. Yeah.
So in what circumstance would that actually happen?
So the plants like rice, wheat, and soybeans do this. So some of those plants in there don't do well in arid climates because of that in there. They're not adapted to it. So if they're in a climate that is not hot and arid, this is never an issue for them. So a lot of the plants here would actually be C3 plants in here. And if all of a sudden it turned into something hot and arid here, it'd kill off a lot of them in there. So that's why you can actually, like, if we have a summer that we have a period of time that's really hot and dry here, it only happens I've seen maybe 2 of them in my lifetime here. So it's not very often. You have a lot of plants die that summer that just aren't adapted to it. So a way a lot of the plants are going to deal with this is the C4 plants in here. They have an alternate mode of carbon fixation. And so what they're going to do is they're going to separate where those reactions occur in there. So if you separate where those reactions occur, you're not going to lose your carbon dioxide. So you can bring your carbon dioxide in, and then you can keep it separated so that you still have the carbons coming in, but you're not going to lose all of your water in there at the same time. And so I'm not really worried about you knowing exactly where in the cell these things move. You've got your mesophyll and your bundle sheath cells in here. So you've got one of the processes occurring here in the mesophyll, and then you've got other processes occurring in the bundle sheath cells. So this is an example of where you're going to have the bundle sheath cells only have photosystem 1, and they'll do cyclic electron flow in there. By having the cyclic electron flow, they don't have to worry about having the electrons go with the hydrogen and losing the water as much. So the CAM plants are another way of dealing with this. And what they're going to do is they're going to open their stomata at night. So they'll take in the carbon dioxide at night, and basically they do their light reactions during the day, their carbon fixation at night in there. So succulents are one example of a plant that does this. Pineapples do, a lot of cactuses do. And so they'll have more of a waxy type of leaf in there that helps to protect it. The what? Yeah. So we have a few of these plants here. The succulents are pretty versatile in where they will go, but the cactuses and stuff are not quite so versatile. They have to stay indoors like that. But that way they don't lose the water during the day in there. And that's also why they have that waxy film on them, is to help reduce water loss in there. So, importance of photosynthesis. It's only the green parts of the plant that do photosynthesis. The rest of it is going to rely on the organic molecules that are produced in the leaves being transported to them. And there, some are going to form sucrose to go in other anabolic pathways. Some are going to form glucose that gets linked together to make the cellulose in there. And they'll just stockpile extra sugars as starch in there. So again, its main thing that it makes is glucose, but just like with respiration and what we eat, it not only being glucose, we eat a variety of sugars that get converted to glucose. That glucose gets converted into a variety of other sugars in there. So any questions on the photosynthesis? All right, if you want to take a few minutes and then